A very good evening to everybody. Welcome to the Different News, another episode once again. So today I have Gopalakrishna Ramaswamy, the founder and CEO of TheraCUES Innovation, with me on this episode. So we will talk a lot and we will have a lot of insights from Gopalakrishna Ramaswamy. Gopi, that's the way we call you. Not
Yeah,
with this.
sure, sure.
Thank you so much for being here and accepting my invitation. And it's great to meet you again after quite a few years. Thank you.
Sure, sure. Same here, Dr. Gargi. Uh, I know last probably 4.5 years before we had a talk to all the dentists. I think you had arranged. That was fantastic. So I'm glad to know that you have started this fantastic podcast. And then I'm eager to know what you're going to ask me and then how I can answer whatever the best I can do it. Thanks.
Yeah, of course. I mean, it's all about your journey. So, yeah, Gopi. So that is my first question. Please tell us about yourself and your inspiration behind bringing up TheraCUES Innovations.
Okay, sure, sure. Uh, See, myself, I have done my PhD at National Center for Biological Sciences and UAS. I'm a plant molecular biologist, basically, but turned into, see, once you learn the molecular biology, you can always do multiple things. So it's not something you have to stick to only one type of the stuff. So I've done my postdoc at Yale University and then Yukon Health Center. The reason why I went to US is to do more of the RNA biology. That was the hot area during the RNA mechanism we were looking at, and then microRNA mechanism. So I went to do my postdoctoral studies. So before that, as an undergrad student, I had written a hypothesis: RNA-dependent RNA polymerase has to be there in all the cells. They will have a normal function. So people are still figuring it out. So when you start studying, you think you're going to win a Nobel Prize. So that is the way we all start uh, trying to do some discovery at that time.
Right.
So time will tell whether you're going to win a Nobel Prize or you're going to do something else. So that's how. While I was finishing my postdoc, again, when I wanted to go out, I always wanted to come back and then do some service to science in India because when I was doing my PhD, it took almost like to get a one kit. It took almost like 8 to 9 months to procure the kit. So that was the time around 98 2000 around that time. So we always wanted to contribute to back to Indian science. So that's how I came back. Then I joined Invitrogen as a kind of a first employee. I was lucky that way. They were setting it up here, and then I joined there, and then I worked with a lot of senior directors, and then who were setting up at the time. So that's how my journey in Bangalore started as a professional. Again, 5 years when I was finishing my one of the as a scientist I was working, and then I was doing certain work. We were doing good, but you get saturated at many places. It's not something like you will continue enjoying everything whatever you do. So we always look for a new challenges at that point. So that's how I moved to the commercial side. I went and uh, joined as a techno-commercial manager at Thermo Fisher at the time. So when I was working there, I got a call from a company called Genotypic. They wanted me to head the business at that point. So if you look at it, a lot of these technology companies, they need to know both techniques as well as the technology you should know, and as well as the commercial part of this. So that becomes a good combination for us to move forward. So again, when I was completing around 5 years, I was working in Genotypic. So as you know, probably 2015, 2016, the genomics information was really exploding everywhere. So it's Now it has become really commodity. So earlier, at least, many people were working, and then I was looking at what else the next challenge we can do it. So one thing is genomics has to take for the translation research. So that's the one thing which I found that as a gap. So when I wanted to start the as an entrepreneur, so that's one thing I looked at it, and then I started a company called TheraCUES. If you look at it, CUES is the signals for targeted therapy. So Thera is kind of a dub for the therapy, targeted therapy. So the CUES is signals. So the combination of this is coded for the targeted therapy. So we work majorly with the translation research companies and then the translation researchers, because we are not going to compete with everyone who is working on the just the genomics area. So that's how the company started. We have been so lucky to have fantastic team we have developed, and then we have fortunate last 7 years. We have almost like August 25th. We are completing around 7 years as an operational. So seventh anniversary is coming up for us. So it's interesting. I know it's not when we started. I never imagined it's going to take so much time to build the company and all those things. But that has been fun, and then it has been enjoying last 7 years. So that's what I can put it in a couple of words.
I know it's a fantastic journey and it's a long journey that you have already, it's not only about the company, but probably you have started longer back when you started thinking about doing something in this area. And congratulations on that you are going to hit your next anniversary in 2025. That's amazing. Um, so, um, Gopi, can you tell us like how, from your perspective, what the work TheraCUES is doing in that perspective? How could we put precision medicine? What would be your perspective on precision medicine that way?
Yeah, sure, sure. I think that's a very good, relevant question if you look at it. So people use precision medicine slightly differently. Also, they also call personalized medicine. So targeted therapy. There are multiple words they use. What I would say is that precision medicine is, see, the diseases are not going to be like so easy in the future. So because we know that especially the diseases like cancer or neuro disorders, or the immunological disorders, all these things, they don't play with just because of the one gene and then one mutation, one thing. So it is a complex of some five six genes might be playing certain times, or this multiple pathways might be having a role in certain diseases. So it becomes very very difficult for people to pinpoint and then say that okay, this is the drug you need to give. For example, the cancer also has there are many cancers are there. So targeted therapy or the precision medicine plays a very very important role. So for example, if you look at lung cancer, you start with one drug, and then when the relapsing happens, there are some mutations have happened, and then you need to identify whether mutation has happened or not, and then you go for another drug. So I'm just giving you the general information. So but you have to give the right drug for the right patient at the right time. So this is very very important. Otherwise you are using a lot of you're traumatizing the patient without providing the proper drug, and then that will also lead to a really huge loss financially, as well as their hopes are also lost because if you're giving the wrong drug. So that is one thing which, that's the way we are looking at how precision medicine could be. We are not actually a diagnostic company at this point. So but we work with enabling technologies to help the precision medicine researchers. It could be clinical researchers or the preclinical researchers, hospital researchers especially, where we work with them to find out okay what types of biomarkers they are looking for, or what types of tumor and tumor microenvironment, how it is interacting. So these are the questions which researchers are groping around. So we are trying to work with them, and then we have helped quite a bit of the researchers, especially hospital researchers, in that direction. So we are not at this point helping the patient, but indirectly through the researchers or the drug companies helping the patient. So that's how the precision medicine we are enabling.
That's wonderful. Thank you so much for sharing your
Sure.
insights. Um, Yeah, so Gopi, you, I think when it comes to novel biological assays, there are always challenges when you develop this kind of assays, right? So,
Yeah.
could you tell us something around this, the kind of challenges probably you have faced, or how, if you have faced, then how you actually overcome them? So maybe this would be a great insight from learning from you.
Sure. 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 Uh, See, for example, when you look at, uh, let's say that you are looking for an interaction of the host and pathogen at certain places. Uh, so when you do certain techniques, either you are looking for one or two genes for the host or one or two genes for the pathogen. So, uh, we have a technology called encounter technology, where I can design up to 800 genes. Either it might be coming from a host or whether it's coming from a pathogen. So both I can look at it and then design as a one assay and then look for those molecules. So that helps me to understand the host and pathogen at one shot. So it's I can understand the what the host is trying to play as well as the what pathogen is trying to play. So can we use a different type of drugs in those situations? So that is one thing which we have done multiple projects in that direction. So there are certain TB works we have done for tuberculosis, where hundreds of genes have been looked into that, and then signatures have been developed for a customer. It's not our own work, but for a customer we have worked with them. So like that, we have around 150 projects we have done. At least 20-30 novel projects we would have done in that direction. So that is a contribution we have made in the direction.
Amazing. And that's very interesting what you said, the kind of test that you are doing where you can actually see both. Basically, you can study host pathogen interaction in one shot. I mean,
Yeah.
that's quite interesting, and very
Sure.
useful information also comes out of this kind of data that we get out of this
And
kind of
the,
test.
the good
Hmm.
thing is this technology, what we are using, encounter technology, within 2-3 days, I can give the result to the team.
Oh
So
wow.
if you use the NGS technologies, it takes 3 to 4 weeks. If we plan well, encounter the one thing is once we have to procure the reagent. That is the time it takes some time. Once you procure the reagents, assays will take 2-3 days. So it's we can give quite quick turnaround time for the especially the preclinical and clinical researchers in that side.
Yeah, that's amazing. I mean, because that's very important when we are into the field of clinical research, because the time is crucial here, definitely.
Correct.
So Gopi, how do you prioritize which area of research to focus on? Because you told that you have worked on so many novel projects. So whether it is like say infectious disease or neurobiology or any debilitating disease like cancer, so how exactly you prioritize which to focus, or I mean, what's the way you deal with this?
Sure, sure, sure. So one is uh, it's easy when customer comes and asks us. So it's, for example, so cancer is the major thing. Many of the projects we have worked. At least 25 to 30% of the project we would have worked on the cancer. Another 20-30% we have worked on the infectious diseases, especially the TB. If you look at it, so the priority here is some of these technologies are highly applicable for the translation research. So if it is not translatable, if they are going only for 4 samples, 6 samples discovery mode, some of our technology might not look good, and then it might not be suitable for the cost wise if you look at it. So the customer will always come back to us the with whom TB, which I'm telling. We have worked with 300, 400 samples. So uh, the cost per sample decreases phenomenally as you look at the infectious diseases. Usually they work on the number of samples are high, because cohorts they can have latent TB or non full blown TB, those kind of things. They can have 300, 500 samples easily. They can collect it. For 100 gene signature, they can look at in the whole blood cell what types of changes have happened. So it is based on the customer and then the technology we match together and then decide what we have to go for. So these two are majorly we have worked on that. Neuro is, there are some 3 4 projects we have done, but there is a lot we can do in the neuro project. So that is the one thing.
Okay, thank you, Gopi, for answering that question.
Sure.
Well, so what do you think, like how cell signaling actually impacts the development of targeted therapies? If I have to ask like a direct way of a question, so can you please enlighten us on this aspect?
Yeah. See, cancer is again a cell division problem, isn't it? Cell division or cell growth problem. Uh, that happens because of the hormonal changes or the hormonal. Either it could be a overexpression of certain hormones might be there, or certain receptors are kind of got amplified, and then that is the reason you are seeing the signal or signaling of certain things. The best case scenario is the EGFR signaling, which is happens in the breast cancer and other places. So we do work with the EGFR positive samples, EGFR negative, and then negative breast cancer samples. All those things we do work with them. So as long as, see, again, we have worked with the pharma companies where they are looking at certain signal molecules they are trying to perturb, so they are trying to stop overexpression of our signaling of certain things. We have found out certain alternative pathways for them, and then they are working on certain drug discovery pathways. So this is the way we are going to help them or enable them. So it's certain drugs where they are acting, whether they are really bringing them down the certain signaling pathways. Those things we can find out through the pathway mechanisms. We have pathway genes called pan cancer pathway, the pan cancer progression, how the cancer is progressing, how the immune and cancer cells are interacting, pan cancer immune cells, immune pathways. So those three gene pathways are the pathway panels we use regularly for the cancer understanding. So again, if they are looking at a drug therapy, whether it is working or not, we have a panel called IO pathway, immuno-oncology panel pathway. So that is useful for the immunotherapy, or if they are trying to work on those things. So these are the way we look at the actual the problem what they are looking at, and then what is their research problem. We suggest them what pathway panels we can use. If some of these things, if they are not able to use, we also do the custom signaling, custom code set we create, and then help them to find out the pathways. So that is the way we work.
Um, so Gopi, coming back, like when you are talking about assays or providing such solutions, particularly in translational research
Sure.
area, one thing that definitely comes to the mind is about the accuracy and reliability of the assays. So if you wouldn't mind, then I ask that how do you ensure the reliability and accuracy of these things that TheraCUES is providing as solutions to the translational researchers?
I think that's a very excellent point. So, when you are doing discovery, a lot of the times people take it for granted. But when you are doing the translation research, you need to have a right controls for all these things when we are looking at. The good thing about the technology assays what we have, it's called encounter assays where we regularly do. It has internal controls called uh, positive controls and negative controls in each sample. Or if for some reason if the signal is low in certain things, you will be immediately you will come to know what changes have happened in that. So if your genes are uniformly is not expressed, these positive and negative controls and the housekeeping genes controls, so we take a look at that carefully, and then if it is going beyond the permissible deviation, we will request to repeat those assays, or the samples have to be relooked into that. All those things we take care of that. So the good thing about all these assays are internal. Lot of controls have been internalized indirectly, and then as we are doing the analysis, immediately we'll come to know where the problem is. So that is the one way we can sort out the things, and then we make sure that it sticks to the QC's parameters. So, for example, RNA, if it is degraded, especially in the cancer, what happens is people keep the FFP samples, and then after retrospective studies they do. So there is a thing called DV200 for each RNA. We look at it, and then we change the amount of the RNA based on the DV200. So there are certain inputs, values we have to give, and then we work on it. So all these things are taken care just to make sure that we give the right data to the client at the end of the day. So that's
Thank you. Thank you so much for answering that question. And I think you are doing a wonderful work for the people into the space of clinical translational research. We are almost at the end of our conversation, though I don't want to let you go. I want to talk more and more, but yeah, we have to draw a line of time. So, Gopi, what are your, with this journey of TheraCUES, what are your future plans? I mean, whatever you could share in your permissible way, uh, that how, what are your chances of, I mean, what are your ideas of expansion? What more you want to do? What more you want to contribute to the scientific community?
sure,
That
sure,
would be
sure.
very nice to know.
That's, thank you for asking that question. So it's so as earlier I was when I was explaining how I started the company, we always ask what's next. I think that's uh, one of the Samsung's ad also comes like earlier it used to come. What is
Right.
next? Kind of thing. So it's we always should look forward in a company where growth is a key thing. So so we are all technologies. We are not like really business people. So at the end of the day, we make money to manage the provide comfortable thing for the employees. Then it's also we look at every day. We look for the new challenges. So as we started 7 years before, we started with one technology called the encounter technology, which we really popularized in India for nanostring technologies. And then last couple of years before, we embarked into another platform called GMX DSP. That's where the digital spatial profiler. So you can take a tissue section and then look for the given region of interest and then see what changes have happened in the gene expression or the protein expression. So it could be anywhere between 1,800 to 18,000 genes you can look for in those places. That's where the tumor and tumor microenvironment we are working regularly. We think that is the very crucial thing for the immunotherapy. So we are looking at developing certain products in that direction. We are also looking at how do we go to the next level. So it's what we are looking at is a group of cells. We are working on with certain initiating certain partnership or technology partnership with looking at how do we look for the single cell spatial. So it's that is the level of the scientists can go, isn't it? So it's from organism, if it can go on in a single cell, what changes have happened, and then if I have a small signal in my brain, what changes are happening in my nerve in the foot. So this is the kind of a thing which people are looking at they are looking at already, and then we are trying to embark in those technologies. So single cell spatial and then single cell technologies, single cell sequencing, these two things which are on the card. We are also looking at how do we reach out to multiple people. So we are looking at partnering with multiple companies, and then how do we work with either channel partners, either in India or globally. We are open to multiple partners. At the same time, we have to have a match between the partners also. It's not something which everyone can do everything. So since it is slightly highly technology oriented, we are looking at how do we work with the right partners in this direction. So the next couple of years, we are going to be working in the single cell spatial, single cell sequencing in this direction, and also the tumor and tumor microenvironment product development. So these three, four things are in the card. So that's going to keep us busy. But at the end of the day, next 3 years, can TheraCUES can grow to 100 people company? So that is the one question I keep asking, because when I always wondered how, uh, if you look at it, Bill Gates and these people, what kind of visions they had for to create 100,000 people company, or even Narayan Murthy and all these people. So it's amazing to manage or the build those kind of enterprises where a lot of things could be managed by the team. So I know big is always sometimes scary also. So it's not so easy to manage. So that's where I'm looking at how do we get certain mentors, certain partners, how do we build a strategic partnership and then grow in that direction. So first thing would be 100 people, and then next 1,000. I hope I'll be able to see those things next in 5 6 years. So that's what.
Absolutely, absolutely. You will be able to see, and probably when you get that mark, or before that, again we will meet and again ask you how was your journey. So that's
Sure, sure.
always interesting. I mean, science is always intriguing and it is always
Yeah.
fascinating. And we just need to, just like as you are committed to your work and journey, we just need to do that. That's the duty of a scientist, because as you rightly said, at the end of the day, we are bound to science, and definitely we need money to grow the business, and create opportunities for people to work as a team. The last question, but not the least, to you is, how do you, as an entrepreneur in the biotechnology field, how do you foresee the growth of bio entrepreneurship in India?
Sure. I think we have a lot of potential if you look at it. At least uh, if you see, uh, compared to last 10 years, we are much better. I know sometimes people have this illusion like everybody think they're going to make billions of dollars as soon as they become entrepreneur. It's not that so easy for young entrepreneurs. I'm just telling. So it is a journey which is you have to live every day, morning and night. It's many of the times you will not have support to do anything. So it's you need to be aware of, uh, if no one is there, can you still move the card or move the cart. So this is one thing they always should remember. But as many of the great leaders would have said, perseverance is the key thing. If you look at it, if you don't have perseverance, it's very easy to drop off. And then probably if we all work for others, we will probably would earn much more money and then peace of mind. But as an entrepreneur, it's a different bug altogether. So it's not for money. A lot of the times people do. It's for the passion, and then it's for the contribution what they can do to leave some legacy in the thing. So that's what all the entrepreneurs would look at it. I know biotechnology or the bio entrepreneurship is kind of merging quite a bit with the IT, or the now that AI and ML is heavily on the multiple domain. It is already even in the drug discovery and as well as the biomarker discovery, all those things. I see huge potential in at least merging certain platforms or merging certain domains in this area. So that looks exciting for us, and then we will see how it can we can take it forward. And then I always feel I should have been 20 years younger now. So that's that's the way I look at it. So it's having said that, so it's have enjoyed whatever we have done. It's I see youngsters have more potential to do that. So that's what I can see.
Yeah, thank you so much, Gopi, for being here. And it was fantastic talking with you. And I wish you all the best. And definitely, very quickly, you will hit the mark of 100 people, a team of 100 people. And we all look forward to that day. And thanks again. Thanks a lot.
Thank you very much, Dr. Gargi. So I know you're doing great job. So I look forward to meet you sometime. So thanks a lot. Bye.
Definitely. Definitely. Thank you everyone for watching this episode, and don't forget to subscribe the channel, or if you're watching on LinkedIn, then definitely put your comment out. And if you have any questions, or if you want to connect to Gopi, where can people connect you? If you just like to share that, it will be great.
Yeah, sure. So my email ID is gk@theracues.com. So my phone number is 98805222715. Anybody can message or reach me out. So thanks a lot.
Yeah, thank you. Thank you everybody. Stay well, stay safe. Bye bye.